just the first part of the experiment. So up until you're going to prepare a column when you're reading the experimental. But do the pre-lab for the whole experiment for this week. So when you come to lab, we're going to check for your pre-lab for the entire experiment. You'll have quiz number 2 this week. Next week, we'll finish experiment 17. And then remember, experiment 16 and 18 are due in lab next week too. So make sure to get your GC mass spec data from your lab prof, and we'll process your NMR data this week in lab for you. And then here's a reminder, open lab hours Monday, Wednesday, Friday, 9:30 to 3:30. Okay? So we're going to start with experiment 17, and then at the end I'll give you a couple notes too for experiment 18. So in experiment 17, we're looking at the Wittig reaction. And we capitalize it. It's after someone's name, the person that discovered the reaction. If you look in, so I don't think you're quite to this point in lecture yet, but chapter 19 in McMurray covers the Wittig reaction, or it's discussed in chapter 19. So if you want to look at it in your textbook as well. But the basic reaction is you're going to start with what is called an ylid. And the Z and the Y here are just substituents off our ylid. And this ylid has a positive and negative charge. So you've got two charges, positive and negative, adjacent to each other with the ylid. And this will react with either an aldehyde. It reacts best with aldehydes, but it'll also react with ketones. So you could, this general structure here could be either an aldehyde or a ketone, with one of the R groups being a proton. And this reaction is really useful because what you end up doing is forming a carbon-carbon bond. You form a carbon-carbon double bond, so even better yet, because then you can do even more chemistry off of that double bond. So this is a really useful reaction that is still used quite a bit industrially because it's so practical and it's a way to form that carbon-carbon bond that isn't so easy to do. And so what you get at the end of the reaction is this, with that double bond in the center. Now, the one drawback of this reaction is you do form, so that our triphenylphosphine has to go somewhere, and our oxygen has to go somewhere, and they end up going together. And so what you get is triphenylphosphine oxide. And so this is a bit of a bugger to get rid of, and so that's the one drawback with this reaction. So there's been, since the development of the reaction, there's been other modifications made to the reaction to try and get rid of this triphenylphosphine oxide. Okay, and so we're going to do just a regular old Wittig reaction with no modifications this week. So we will be having to get rid of the triphenylphosphine oxide. Important thing here, this is a neutral compound because you've got a positive and negative charge, so a dipolar compound. But again, those charges are on adjacent species in the compound itself. Now, what you can do to form the ylid is actually start with a salt. And so if you make the triphenylphosphine salt, and then, so this is the positive charge, and the counter ion has the negative charge. And then you react this with a base, then you can form the ylid. And so that's what we're actually going to do this week, is start with a salt and then make the ylid before we go on and react it with the aldehyde. Now, what's really nice about this reaction too is making this salt is really versatile. So you can start with primary or sometimes secondary alkyl halides will work, and you react it with triphenylphosphine, and then use a base, and you can make this salt. So you can make the salt of whatever you want to react with your aldehyde and ketone, or ketone, as long as it's primary, or some of the secondaries work pretty well. Okay. Now another thing that we talk about with ylids is something is either stabilized or not stabilized. And so when you have your Z and your Y is either an alkyl group or just a proton, we talk about that being unstabilized. And so your Z and your Y are alkyl or just a regular proton. And so what that means is we can't just form that ylid and it'll stick around. What you have to do is form it in situ. So you have to start with the salt, and right before you do the Wittig reaction, form the ylid, and then quickly use that ylid to go on to the reaction. And so these guys are not very stable to air or water. Okay, so you have to be really careful with the unstabilized. Now the stabilized ylid, you can just put it in a bottle. They'll stick around. And what you want is your Z and or Y to have an electron withdrawing group, or withdrawing groups, and that will help stabilize it. So think about the positive charge is always going to be towards the phosphorus. The negative charge is going to be on the carbon adjacent to the phosphorus. So if you've got an electron withdrawing group, that's going to help stabilize that negative charge and help make it more stable. Okay? And so then you can form the ylid. It'll stick around. It won't decompose on you. You can make it, and if it's like a solid, isolate the solid, use it later, things like that. So what we're going to do this week is we're going to actually start with the salt and make the ylid in situ, but our ylid is stabilized. It's going to have an electron withdrawing group on it, so it will last a little while longer for you guys and not just be so reactive that it disappears on you. So with these ylids, there's resonance structures that you want to look at too when you're looking at the stabilized versus the unstabilized ylid. So if we look at, and this is in your lab manual as well. So here's an example of an unstabilized ylid. We've got a proton here and some sort of alkyl group here. So here's our ylid. Another way that you can write it, another electron or another resonance form, is then to put the double bond here. Okay. However, when you have this is the ylid that we're going to use for our reaction. When you have a stabilized ylid, you've got these two forms, but also you can have resonance with that electron withdrawing group that is off that carbon as well. Okay, and so then the more resonance you have, just like with anything else, the more resonance structures you can draw, the more stable it's going to be. Okay. So just know between these two, we've got two resonance structures here. We've got three here because of that electron withdrawing group. Okay. And so, and like I said, this is straight out of your lab manual. I'm going to put this up here and then also go through drawing it on the board for you. So this is the first part that I've showed you. We can use a base to take our phosphonium salt and make the phosphorus ylid from it. So just forgot to put over here some sort of base. Okay. And so if we do that, and we have our ylid, and it's then going to react with an aldehyde or a ketone. The negative charge is what's going to attack this carbonyl. And so, have you guys started alcohols in lecture? Okay. After alcohols, you're going to go into carbonyl chemistry. So you'll learn more about attack of a carbonyl. So we're going to attack the carbonyl of the aldehyde or the ketone. And then what we're going to do is eventually form our alkene. But our intermediates that are involved are shown below. So we're going to form what is called a beta-ene. And so this is very similarly related to the ylide, except for we don't have the charges on adjacent atoms. We have two charges involved, but they're not on the adjacent elements of the compound. So we got our negative charge here, our positive charge here, and here's our Y and Z groups from our ylide. What happens is after this attack, this is the guy, so let's show it this way here. I was going the wrong direction. So this is what you form. After these two attack, you form this guy. You're not ever really going to, you're not going to isolate your beta-ene. But what it does is we're going to form a four-membered ring from this. So we've got electron flow that way. And then our four-membered ring is then going to collapse, okay? So here we've got our and so our bond here is going to go with the oxygen and the triphenylphosphine, so that's how we get our double bond there. And then we've got this collapse here. That's how we get the double bond between this carbon of our starting aldehyde and this carbon of our ylide. And so then you get the alkene. Okay? But this beta-ene is characteristic of the Wittig reaction. And similarly related adjacent charges, and here we've got a dipole here, or two charges, but not on adjacent spaces right there. Okay? Everyone good with that? Okay. And so here's page 707 in the 6th edition of McMurray, and 720 in the 7th edition shows this mechanism that I just showed you. Okay? So now let's look at what we're going to be using for your reaction. So this is our salt here that we are going to use. Quite a mouthful to say. It's our, this guy is carbomethoxymethyl triphenylphosphonium bromide. Okay? So that's the guy we're using. We need to form our ylide first. So we're going to use sodium methoxide in methanol. And you want to, when you're doing this reaction, as you go on through carbonyl chemistry, you're going to talk about one of the reactions is transesterification. Okay? To prevent transesterification, and so getting what we don't want here off of our ester, we're going to use the pieces of our ester off of here. Okay? So when you get to transesterification again, or when you get to transesterification, go back and look at experiment 17, and hopefully it'll be clear why we used sodium methoxide in methanol instead of using sodium ethoxide. Would work just as well in ethanol or other bases. Usually it's sodium methoxide or sodium methoxide. But there's a particular reason that we use the sodium methoxide in methanol, so that we don't end up changing the species that we want to react. Okay. So we form our illid. So think about what arrow pushing would be involved here to form this guy. We've got a base. We're going to take our proton. Then we've got negative charge here, forming the illid. Does everybody see that? So we've got a base, sodium methoxide. Take positive charge there, and then remember this is just one of the resonance forms of the illid. So you could also draw this as a single bond, and plus and a negative, right? Okay. So then this guy is going to react with our hydrosemaldehyde. That's the aldehyde that we're going to use. Okay. And then we'll get this product, the cis-trans product of this alkene plus triphenylphosphine oxide. Okay. What do you think the hydrosemaldehyde is related to? Just based on its name. Ideas? Like formaldehyde, something that's going to smell like cinnamon? Possibility? Okay, so this guy doesn't smell so bad. So for once we'll have something, after our stinky unknowns last week, we have something that won't stink quite so bad. It's going to smell like cinnamon. Okay. Now one thing with this reaction, you want to keep things, we do have a stabilized illid, but our base, our sodium methoxide, will also react with water, so we don't want to kill our base, okay? So you do want to keep things dry, keep the moisture out of your reaction, okay? So make sure you're using dry glassware, keep things covered, make sure when you are stirring, you're going to have a period where you're forming this, forming your illid, so you're going to react your salt with the sodium methoxide. Make sure it's covered when it's stirring. And then we're going to dispense the, TAs or the lab instructors are going to dispense the hydrosemaldehyde because we have to do it with a syringe and you need just a very small amount. So you're going to come see us in the hood and we'll give you your hydrosemaldehyde. When you come see us, make sure you keep your reaction flask capped. So keep as much moisture out of the reaction as possible, okay? We don't need to take the measures that we did with the Grignard reaction, but you do want to keep things as dry as possible because you don't want to kill your base and have it react with water instead, okay? Now let's think about the beta-ene that would form between here and here, okay? So it's going to start just like our other guy. We've got an oxygen here. Now what would be off this carbon? Proton, yep. And then what else?
The CH2 phenyl.
Yeah, so I'm going to try and write it here so it doesn't get in the way of things, okay? Then this is connected to another carbon. And then what's going to be off? So we've got our negative charge here, triphenylphosphine. What's going to be off of that carbon? Which carbon is that guy? So this part, which carbon is it? This guy here, right? Okay. So you're going to again have a proton off of it. And then you also are going to have your ester, your electron withdrawing group. So, okay, and positive charge here. So we've got that, and we go on form our cyclic intermediate. Okay, so then collapse is going to work just like we did over here, where we go like this, go like that, and so from that, can you see how that product is formed? Do you see this guy from that collapse? Okay. So you're going to go on and form, I'll draw it just like it is. So we've got this carbon and this carbon are where your double bond are. And then we show the protons like this because you're going to get both cis and trans product. Okay? And so this is the carbon-carbon double bond that formed from the reaction. And then just like before, we're also going to get a triphenylphosphine oxide. Okay. Now one thing with stabilized ylids, they have a tendency to make more trans product than cis product. You will see both cis and trans with this reaction, okay? Don't be surprised if you do see more trans than cis, but you will see both cis and trans, and you can actually, we're going to monitor the reaction with TLC, and then we're going to, we're going to look at your final product by TLC, and then you're going to separate out your product mixture with column chromatography, which is, I'll talk about next week, also related to using TLC. So you can, when you isolate your product, you can actually see cis, a spot for cis and a spot for trans. You can see both of them, okay? But typically with this reaction, you're going to go more towards trans than cis. You can actually, with the stabilized ylids, you can even, with the right combination of base and combination of ylids, you can really push the reaction towards the trans product if you work out the conditions just so. So something else to know about the stabilized ylids. This reaction, so I talked about you're going to take your salt, you're going to, first you need to dissolve it in methanol, then you're going to add the sodium methoxide. You're going to use a transfer pump like is used a lot in Gen Chem to dispense the sodium methoxide. It is a base, so be careful, wear gloves, don't get it on you, okay? And so you're just going to pull up on the plunger and then squish it down into your reaction, okay? Or if you want to put it into a graduated cylinder and then put it into your reaction. Either way. Then after you've stirred your methoxide in your salt for 5 minutes, you've formed your ylid, then you're going to carry out the reaction. You'll get your hydrosamaldehyde, and then you have to reflux for an hour. And you need to do it in a hot water bath. Okay, so you're refluxing for an hour in a hot water bath. That means start with hot tap water, but also make sure your flask is down in the bath and your bath is full of water, so you don't have to add cold water. Stop the reflux, wait for things to heat up, get the reflux going again. Okay. During this time, for the most part, except for Thursday morning lab, it'll get a little bit interesting because we have 2 labs going on. We are going to teach you how to use TopSpin, which is the processing software for processing your NMR data, okay? So you're gonna set up the reflux, then we're going to have the TAs watch the reactions, and you're going to go learn how to use that processing software. And by the time you leave lab this week, you're going to have your NMR data that you need for your unknown for experiment 18. Okay? So, especially since you're not going to be watching your reflux, make sure you have plenty of hot water, make sure it actually starts refluxing before you leave it, and keep track of the time so we can let the TAs know when an hour is over. Okay? Then, after you reflux for an hour, you need to get rid of the methanol. Well, it's already good and hot from refluxing for an hour, so we're just going to distill the methanol, okay? And you're, again, going to use a hot water bath. So we're not using a heating mantle at all during this reaction and during the isolation of the product. Then let it cool completely, okay? Because then the next step is precipitating out the triphenylphosphine oxide. And so you're going to distill off the methanol. Then you want to make sure you cool that flask completely to room temperature. So you then you're going to add hexane to it to precipitate out the triphenylphosphine oxide, okay? Because the triphenylphosphine oxide is very polar. It likes the methanol. We're going to add hexane and just a little bit of methanol to it to help precipitate out that triphenylphosphine oxide, okay? So what's going to appear in your flask is kind of an off-white solid. Don't mistake that as your product. So your product is a liquid. The solid that you're forming is the triphenylphosphine oxide. Okay? And we'll use gravity filtration, get rid of the triphenylphosphine oxide. You may have to do it a couple times. You want to get as much out of there as possible. Because next week what will happen is when you run the column, that solid kind of just crystallizes out on the top of the column, and it'll make your column run slower and kind of streak through your column next week, okay? So it won't give you as good separation next week. So try and get out as much as possible this week. If you filter once and have solid, filter a second time, get as much solid out as possible, okay? Then what we're going to do is we need to remove the hexane. We're going to remove the hexane with what is called a rotary evaporator. So all of a sudden, you're going to get really upset that we ever made you distill anything, because you're going to learn how to do it really quickly. Okay? But we needed you to learn how to do it, and there is times that you actually do have to distill and you can't use the rotary evaporator. So this is kind of generic picture of what one looks like. So we're going to teach you how to use it. So under vacuum, you're going to remove the solvent. So this works when a simple distillation would work to remove a solvent. Now when you're doing a distillation to purify something or a distillation to collect fractions, you can't use a rotary evaporator. So that's why you had to learn how to use a distillation. We have you distill the methanol off because you already have it hot. You already have the water bath hot. Everything's, if it's refluxing, it's really quickly and easily going to convert to a distillation. Just remember, be careful because you're handling hot glassware with hot vapor. But you can quickly convert the reflux to a distillation, and that's faster than everybody trying to use the rotary evaporator, okay? But then when you get to this point, things will kind of be staggered out depending on how things went with precipitating out the triphenylphosphine oxide. So then we can use the rotary evaporator. So we've got 3 of them between the 2 labs. Every lab section but the Thursday mornings, we'll get to use all 3. The Thursday morning we're going to have to share between the 2 of them. But hexane has a low enough boiling point, it should come off pretty quickly, okay? So then you're going to remove the hexane, and then you're going to store, you're going to have what looks like not a whole lot of product in that round bottom flask. Store that flask, cap it, put a little bit of grease on it to keep it in your flask, and store that till next week. And then next week we'll use column chromatography to purify it. Okay. Before you use the rotary evaporator, what you'll want to do is you need to sample that reaction mixture. So right here where you still have hexane and a little bit of methanol, right here you want to spot it for TLC, okay? Because then you don't have to dissolve a little bit up in solvent to spot it. So spot it before you rotavap. There are standards for the product and there's standards for hydroformaldehyde in the lab. So you're going to spot versus those standards, all right? Run the TLC just like you did last semester. Keep that TLC till next week. So remember, you're either going to put it into your notebook or you're going to draw a life-size version of it in your notebook. But regardless, hang on to that TLC plate until next week because that's your reference for what everything's going to look like for running your column, okay? Then the other thing with using the rotary evaporator, it's always good to know the weight of your flask before you put it on, so then you can weigh it afterwards and see how much stuff you have. So going into the column, you want to know a crude mass before you isolate it with the column. So with the rotary evaporator, use a tiered flask. You know what the weight is, right? And then, like I said, we'll store it until next week. Okay? Questions with anything with experiment 17 before I go on to experiment 18? Yeah.
Is the quiz over part A and part B?
It'll be over part A, and then next week could be either. But this week it won't cover what I haven't covered in the lab lecture. Yeah. Okay. And safety is always fair game. They could even throw something from experiment 16, experiment 18 on there too if they wanted to. Okay. Other questions? Okay, good with 17? All right, so now a couple notes about experiment 18. First of all, you're going to need, if your lab prof hasn't done so already, they're going to tell you what you need to do for your NMR table. So just like IR, you will need an NMR table, and your lab prof will tell you what that table is going to look like, okay? I want to remind you, this is the example from last week, remind you whenever you are analyzing NMR data, you always want to draw the structure on your spectrum. So you're going to have a spectrum that looks like this, the whole NMR spectrum of your compound. Draw the structure, label the unique protons with letters, and you're going to want to put those letters above the protons. Okay, so this is common. Pretty much every lab prof wants you to do this. So just remember this for the analysis of NMR data, whether it's experiment 18 or any other NMR data for the future. Okay. Couple other things to keep in mind with experiment 18. Like I said earlier, you're going to get your mass spec from your instructor. All of them have now been run. We're going to, I've talked about, you're going to get your, or process your NMR data, so you'll have your NMR data. One thing to keep in mind with that is everybody's going to have a peak at 7.27, 7.26, 7.27 parts per million. What is that peak? That's the chloroform, but which form of it is it? It's the CH, right? So in that CdCl3, there's a little bit of CHCl3, and that's going to be with about 7.27, 7.26 parts per million. So keep that in mind in your spectrum that you're analyzing. And some of you will have peaks that overlay it, and some of you won't have peaks. But make sure if it is visible amongst the peaks that you can identify what that peak is, okay? And it's always the pronated form. So people want to label this usually as CDCl3. It's not CDCl3, it's CHCl3. It's in the CDCl3, okay? Remember that the Aldrich Spectral Viewer and other good websites for getting literature data for experiment 7 or experiment 18 is on page 1616 in your lab manual for good websites and databases. And your data, your IR, NMR data, GC mass spec data can come from all, like you don't have to get everything from one database. It can come from, you could get all 3 different pieces from 3 different databases, okay? So make sure you're checking all of those. Like I mentioned, the NMR data table, make sure you get that information from your lab prof. Remember the unknowns you should be able to find in the back of your lab manual. If you can't find them in the back of your lab manual, then in 3123, so the IR room, is the tables, this one book for identification of organic compounds. And remember, it's kind of falling apart, so be careful with it. Okay. Other questions you had with 18 at all, or 16? Yes?
Was 16 the one that you needed to go actually to the science center to look up the values?
Yeah, your homework out of 16, so with the assignment that you're going to turn in for experiment 16, those values you have to use Aldrich Spectral Viewer, okay? So that's in the Duck Computer Lab, but that's also in any computer lab that's a campus clone computer lab. So the general computer labs on campus should have Aldrich Spectral Viewer on them, okay? If you go to all programs under chemistry, you'll find Aldrich Spectral Viewer, all right? Other questions? I have one more note about experiment 17, but I want to make sure I've got experiment 18 covered. Okay. Everything good? Okay. All right. One thing I forgot to mention with our product for our side product for experiment 17, this solid, the triphenylphosphine oxide, where does this go? It goes in the organic waste. So it's a solid. You're going to rinse it into your organic waste container with acetone. It will dissolve with acetone. So dissolve the solid with acetone into the organic waste. Rinse the filter paper because it gets a little bit stinky. If you still have stinky filter paper, then we'll stick it in a container in the hood, okay? So if you don't throw stinky filter paper, just like, you know, stinky garbage shouldn't go in the outer garbage that's going to make the lab stink. So if you have stinky garbage from this lab, stick it in a container in the hood and we'll take care of it. But we don't want to stink up the whole lab. And the filter paper will have an odor on it, okay? So don't just throw it in the garbage. Make sure it goes in the reagent hood. There'll be a container for it, okay? Any other questions? Okay. You'll get your bonus back. I went over last week, so you get a few minutes back this week. Okay. I'll see you next week.